Now that we've looked at the participants in DNA synthesis, let's look more closely at the process itself. We've seen that DNA polymerase must have a 3' OH on which to build the new polymer. This necessitates that once the replisome has formed and the strands have been separated, the process must begin with the synthesis of this RNA primer. The enzyme that catalyzes this reaction is primase, which must therefore be an RNA polymerase and capable of de novo synthesis. The primosome is the complex that forms at the replication fork to initiate synthesis and contains primase, the RNA primer, and helicase. Note the distinction between this and the replisome. The latter also contains the replicating enzyme, DNA polymerase. DNA polymerase III, pictured below as the blue and purple donut-shaped structures, begins DNA synthesis by adding deoxyribonucleotides to the 3' OH of the RNA primer. As we've seen, synthesis proceeds in the 5' to 3' direction on both strands. As the replication moves downstream, the RNA primer is removed by DNA polymerase I, pictured as the purple donut below, which then replaces the primer with DNA. Notice the yellow donut-shaped structure, representing DNA ligase. This enzyme is responsible for linking the Okazaki fragments in covalent phosphodiester bonds. Pictured here is a ribbon diagram representing the crystal structure of DNA polymerase. It is a complex enzyme with multiple domains, each serving a distinct function. All known DNA polymerases, while serving distinct functions, all seem to exhibit a common structure, which resembles a right hand, with domains identified as the fingers, palm, and thumb. The active site of the enzyme lies in the crevice of the palm, between the fingers and thumb. The fingers are responsible for deoxynucleotide recognition and binding, and the thumb is responsible for DNA binding. By examining the figure below, you can see that there are 3 pol III enzymes bound at or near the replisome. Note that there is one enzyme bound to the leading strand and 2 enzymes on the lagging strand. Initiation of a new Okazaki fragment requires association of a new pol III enzyme. Shown here are the many parts to the replisome, including primase as the green shape and helicase as the orange spheres. The sliding clamp is a dimer of beta subunits of pol III that forms a closed ring around the DNA. The clamp loader is the portion of pol III that opens the sliding clamp and inserts the DNA. It is a pentamer of 5 subunits and uses the energy of ATP hydrolysis to drive this process. This is an excellent example of the relationship between quaternary structure and enzyme function.